Okay, so good morning, children. Yesterday we were doing ribosomes. So I told you that ribosomes, they have two subunits, and both the subunits, they lie in the cytoplasm and they lie in the cytoplasm separately, but they unite during translation. They unite during translation. And for their union, so that they attach to each other, magnesium ions are required. So magnesium, they unite the larger and the larger subunit uniting with each other. This is how they unite, and they unite only in the presence of magnesium ions. They unite, and then there is one more thing which joins to it. They have different functions. The messenger RNA joins to it. This is messenger RNA, which joins here. This is the function of the smaller subunit to attach with the messenger RNA, whereas the larger subunit, I told you it has three sites, 1, 2, and the third site. This is A site, this is P site, and this is E site. A site is the acceptor site, P site is the peptidyl site, and E site is the exit site. Besides this, another function of this is it has the enzyme, it consists of the enzyme peptidyl transferase. The peptidyl transferase enzyme is very important. It has two very important functions to do. Number one is it helps in the formation of peptide bond, and the second function of peptidyl transferase is termination. When the complete protein chain is formed, protein of a certain setup, so that is termination of the protein chain formed is also brought about by peptidyl transferase. Now, I told you it has, genetic codons, okay? It has genetic codon, and this genetic codon, or it has a genetic message, coded genetic message rather an appropriate word. It has coded genetic message. Messenger RNA has coded genetic message. And what is the function of this coded genetic message? This has to be, converted into polypeptide chain. Polypeptide chain. What is polypeptide chain? Polypeptide chain is protein molecule. It has to be converted into protein molecule. Protein chain. So it has to be converted into polypeptide chain. Now, as I told you, when this process of translation starts, the initiator codon is always AUG. So this AUG, which is initiator codon, it is, suppose this is the initiator codon, it will start. Okay, I'll make it here. AUG, the initiator codon, AUG. Okay, the next other codons are there. The transfer RNA for AUG. AUG codes for the amino acid methionine. So this amino acid methionine will be brought by a transfer RNA. It will be brought by a transfer RNA, and where it will be brought? this transfer RNA with methionine will come to P. So the transfer RNA for AUG, it comes at, it is accepted at P. At P, none other, all other transfer RNAs which carry the other protein amino acid. Like next we have U. Okay, so next U codes for phenylalanine. U codes for amino acid phenylalanine. This the transfer RNA which brings phenylalanine. I'm just writing the short of it. The transfer RNA which brings the next amino acid will be accepted here at the A site. It is only the initiating codon, the transfer RNA carrying the methionine, which is accepted at P site. Rest all the transfer RNA which are carrying the amino acids are accepted at A site. So this happens. This is the function of this ribosome, and ribosomes are also known as protein factories, and they help in the synthesis of protein. How they help? See, this has a genetic message which is coded genetic message. The coded genetic message. This coded genetic message is in the form of triplet codon. Okay, and it attaches, this messenger RNA attaches to the smaller subunit, whereas the larger subunit, it has three sites. This is the A site, P site, and E site. The P site is the peptidyl site which receives the transfer RNA carrying the amino acid methionine for the initiator codon, and rest of the transfer RNA bringing other amino acids are accepted by the acceptor codon. Okay, and besides this larger subunit also has the enzyme peptidyl transferase. There are two functions of peptidyl transferase. Number one is formation of peptide bond. Number one is formation the first function of peptidyl transferase enzyme is formation of peptidyl transferase, and the second function of enzyme peptidyl transferase is termination of polypeptide chain. That means when one protein molecule is formed, it is terminated, it is separated from this setup, and that is also done by peptidyl transferase. So this is the function, and this is the ribosome unit, which is very important, both the larger and smaller subunits, and this is the ribosome, which is very important to carry out the function of translation. Now we start up with the process of translation, which goes like this. So here, we start with the process of translation. First of all, where this process takes place. Transcription, it takes place after translation takes place after transcription. That means it is followed by translation. Translation is followed by transcription. So after transcription, translation takes place. The site of translation is cytoplasm. It takes place in cytoplasm, whereas transcription takes place in the nucleus. Now, what is this basically? This is the process by which the coded genetic message, okay, which is brought by or carried by messenger RNA, is converted into polypeptide chain. So it is converted into polypeptide chain. What is polypeptide chain? One amino acid connected with another amino acid, connected with another amino acid, connected with another amino acid. This is one polypeptide chain or protein molecule. This is one polypeptide chain, or we can also say it is a protein molecule. These bonds are peptide bonds. Peptide bonds. These bonds are peptide bonds. And these are amino acids, which are all the amino acids are linked by peptide bonds. And who brings, from where these amino acids come? The transfer RNA brings these amino acids. The amino acids are brought by transfer RNA. So here peptide bonds join the amino acids, which are brought by transfer RNA. And this whole formation of peptide bond in between the amino acid, this whole takes place in the ribosomes. Okay, so let's start. We start with the machinery for translation. What is the machinery required for translation? Machinery for translation. What is required for translation? The machinery for translation includes transfer RNA, that is tRNA. Of course, why this is required? tRNA is required to bring amino acid, because this is the pulley which brings the amino acid to the ribosome where the polypeptide chain is formed. Then we need the coded message, which amino acid has to be brought. That message is coded in messenger RNA. So we need messenger RNA. Besides this, all this takes place in the ribosome. So we need ribosomes. And for attachment, of course, magnesium is required. Then we need amino acids, which are brought about by transfer RNA. So amino acids are also required. Besides amino acids, one enzyme which is very important is amino acyl tRNA synthetase. So one enzyme is also required, and the name of the enzyme is amino acyl tRNA synthetase. This is the enzyme which is required, and this is the complete machinery which is required to perform the process of translation. Now, what happens? What is the mechanism of translation? We start with the mechanism of translation. How this process is carried out. Mechanism of translation. So we are doing the process of translation during which the coded message, which is on messenger RNA, is converted into polypeptide chain. That means if it is a cell, this is the nucleus. Here transcription took place. I'm writing TRCR, transcription took place. A messenger RNA was formed. This messenger RNA, which was formed as a result of transcription, comes out the nucleus into the cytoplasm. So this messenger RNA comes into the cytoplasm. In the cytoplasm, there are smaller subunits, the larger subunits, smaller subunits of ribosome, the larger subunit of the ribosomes, all are there. So what happens? They unite, both the larger subunit, the smaller subunit, and the larger subunit unite. When the smaller and larger subunit unite, they form a ribosome, and the mineral which is required for the union of these two subunits of ribosome is magnesium. So in the presence of magnesium, both of them, they unite, and then what happens? This messenger RNA, it goes and binds to the smaller subunit of ribosome. So here it comes and binds. Now when it binds, it starts reading the coded message, which is in the form of nitrogenous bases. And on that basis, what happens? The transfer RNA is brought here. Okay, to the first one, to the P site, then acceptor site, and here this is the exit site. So first one, AUG, the methionine is brought here. Okay, then the next transfer RNA is brought, which will carry the next message. Suppose it is leucine. Okay, so next will be brought here. So in the acceptor it has come. Now which enzyme is present in this larger subunit? Peptidyl transferase is present. This peptidyl transferase will make bond between the two. This, I mean, methionine and then leucine. And then what will happen? Translocation will take place. And as a result of translocation, the ribosome will move towards This is 5' end of messenger RNA. This is 3' end. It will move towards this side. As a result, when it moves towards this side, the methionine which was present on P side will shift towards here, will shift here to the E side. Okay, and the one which was present on A, leucine, it will shift to the P side, and this will be vacant now to receive. A will be vacant to receive another transfer RNA. So suppose the next transfer RNA brings tyrosine. It is bringing tyrosine. So it will be accepted by this one. And then what will happen? Again, translocation will take place. And as a result of translocation, what will happen? Already the methionine which is there, it will shift up. Already the bond is already formed now. Then the leucine will be here at E site, and the third one, which was tyrosine, it will come to P site. Okay. Okay. Then again it is vacant, ready to receive another, and it will shift more towards this side. That means it will keep on shifting like this. It will keep on shifting like this towards this side. So if these are 3A, P, and that side, so when it moves like this, when it moves, messenger RNA moves, it will keep on moving. So when it moves like this, these things, they come towards this side. They will keep coming towards the translocation takes place. And as a result of translocation, it will move towards next. And finally, a chain of peptides, polypeptide chain will be formed like this. Okay, a polypeptide chain will be formed like this. I'll make it here. A polypeptide chain, long polypeptide chain will be made formed here of different amino acid, and all will exit through the exit site. And then peptidyl transferase will terminate, and a long chain of peptidyl transferase will get terminated from this setup, and we will get a protein molecule. Little, could you understand about the mechanism of translation? Could you understand little bit how a polypeptide chain is formed? Now I'll tell you step by process if this is understood. Yes, beta, could you understand it? Which I told you in the diagram. Yes, bache? Okay. I'm getting the message that they are able to understand. Yes, Paras is saying yes. No other message? Okay. Repeat. Fine. Fine, I'll repeat it. First, I'll not tell you stepwise. I'll tell you what happens during the mechanism of translation. How this translation will take place. This I'm going to tell you. What happens? This is the cell. In the cell, this is the nucleus. In the nucleus, by the process of transcription, a messenger RNA is formed. This is messenger RNA is formed, which is inside the nucleus. Now, transcription has taken place and messenger RNA is formed, which has the coded message, coded genetic message. On the basis of this coded genetic message, protein has to be synthesized. Now what happens? This comes out of the nucleus into the cytoplasm. So this messenger RNA has come into the cytoplasm. Now in the cytoplasm, we have the smaller subunits, the larger subunit, smaller subunits, larger subunits. Now in the presence of magnesium ions, these smaller subunits and the larger subunits join, they combine. So this has formed, this is the larger subunit which has combined with the smaller subunit, and a complete ribosome has been formed. Now this ribosome has three sites: the exit site, the peptidyl site, and the acceptor site. Whereas the messenger RNA, it joins with the smaller subunit. It has a coded message. The first message, the first code is always AUG. It is always AUG. That means the first tRNA which will come to the larger subunit will have the amino acid for AUG, and that amino acid for AUG is methionine. That means, and I told you that the initiator codon, AUG is the initiator codon which initiates the process of protein synthesis. This codes for methionine, and that methionine is not accepted by this A site. A site does not accept it. This methionine is accepted by P site. This is P site and this is P site. So this is accept the tRNA which brings methionine. I'm just making tRNA like this, which brings methionine is accepted by peptidyl site. Now, suppose we have another genetic code, and that genetic code, let it be GUG, GUG, which is GUG, which is for valine, GUG. And next, suppose UUU, which is for phenylalanine. Next is UUU, which is for phenylalanine. So first AUG has been done. The transfer RNA with methionine has reached P. Now what is going to happen? Now the next triplet codon is GUG, and it codes for valine. So next transfer RNA with valine will reach here, and only AUG, the transfer RNA carrying the amino acid for AUG, is only accepted at P site. Rest of them are accepted at A site. So this is A site. The next messenger, the next transfer RNA is bringing the amino acid for the next coded message, which is GUG. So it is bringing valine. So valine will come and it will not attach to P. P is a place of attachment only for M. So it comes and it is received by the A site. So the transfer RNA with valine is received by A site. So it has been received by A site. Now this, when it is received by here, now what happens? The next is phenylalanine. Phenyl UUU, which codes for phenylalanine. So the next transfer RNA will bring phenylalanine from the amino acid pool. Now, by the time what happens, I also told you that the larger subunit has enzyme peptidyl transferase. Peptidyl transferase is also present in this larger subunit. So what it does, it has two functions. One, termination of the polypeptide chain, and the second is formation of peptide bond. Now, where the peptide bond needs to be formed? The peptide bonds needs to be formed between the amino acid. So methionine is one amino acid, valine is another amino acid. So a peptide bond be formed in between the two, but they will, because codes have to be read. So for reading all the genetic code, whatever codes are read, they will shift towards this side, and the codes which are not read yet, they will shift towards this side here, so that the messenger RNA, the transfer RNA comes and attaches to the code of this, the anticodon of messenger RNA attaches to the codon of the anticodon of transfer RNA attaches to the codon of messenger RNA, and then that genetic message is also read. So it will shift towards this side. When it shifts, what happens? These things also shift. They will also shift. A jerk takes place, and because of that translocation, this M, which is M and valine, which are connected by peptide bond, now this M will shift onto the next site, which is M site. So this, the M will shift here, the transfer RNA will M will shift here, and valine will shift to the next site, which is here. Valine will shift here. The tRNA carrying valine will shift here. Now valine has shifted here, so this first site is now empty to receive another transfer RNA with the amino acid. So now the next one is UUU, which codes phenylalanine. So next transfer RNA with phenylalanine will be accepted by this. It will be accepted, carrying phenylalanine will be accepted by the acceptor site. And what will happen soon? A bond will be formed between these two also. So phenylalanine, between phenylalanine and valine also the peptide bond is formed. Okay. Now again translocation will take place because the next has to be read. Suppose it is G, suppose let it be another, say CUU. Next is CUU. It has to be red. And this codes for leucine. So it has shifted here now. It will shift here because this has to be red. So AUG will come here. AUG, GUG will come here. And UUU will, AUG, GUG come here. UUU will come here. And at the same time, when it shifts here, what will happen that this methyl alanine and valine, it will come onto the exit side. It comes after translocation. This methyl, methionine and valine, it comes to the exit side and they go out. The transfer RNA without the amino acid goes into the cytoplasm, and the amino acid, they, because they are bonded by now peptide bond, they form a chain like this. And the one which was here for the next one, CUU, that is leucine, it will be accepted, and after accepting, it will move towards the P side where the peptide bond will be formed between this and leucine. Another chain will be formed, and it is ready to accept another amino acid. And this way, the complete aminos had been used here for forming polypeptide chains. They move into the cytoplasm and they bring another amino acid. They are ready to bring another amino acids. Okay, and thus what do we get? Finally, we get here a chain, a polypeptide chain like this. We get a polypeptide chain with all the amino acids in the, which is a protein molecule. Is it clear? Is it clear, children? Process of translation is clear, beta? Is it clear? Could you understand? Okay, yes. Sukh Sahaj is saying. Amit Katoch, any problem in this? Any question? Any question? Amit Katoch, I'm asking you, bache. Any problem? Otherwise, I'll start with the stepwise process of the mechanism of translation. Any problem in this? Anything you want me to repeat? Yes, bache? Yes, beta. Yes, Amit Katoch. Anything which is not clear in this? Yes, bache? Anything you want me to repeat? Vanshika? So finally, what we get is a protein molecule. This is the protein molecule we get, and this complete process of the formation of protein molecule takes place. This is what this process is all about. Now we are left with 1 minute, and if I start the process, stepwise process, I'll not be able to finish it today. So stepwise process I'll start tomorrow. Right now, I have told you the what about the process of translation, the complete mechanism, how it takes place, the machinery for translation, the mechanism of translation, the machinery which is required, the introduction of the mechanism of translation. Children, see these terms, they, when you hear them, you find them difficult. Like polypeptide term, you all find difficult, but what is it? It is just amino acids which are attached by peptide bonds. Polypeptide. See, when I write here, coded genetic message carried by messenger RNA is converted into polypeptide chain. Coded genetic message, that means the messenger RNA, it has all the bases in it, and these bases, they are carrying some message. What message they are carrying? Suppose it is carrying AUG, that means amino acid will be brought. Then UUU phenylalanine, GUG valine, GAG leucine. Okay, CUU leucine again. Okay, valine, leucine. These all This is the messenger RNA carries the coded information. This is messenger RNA, which is carrying the coded information. So coded genetic message carried by messenger RNA. This messenger RNA is carrying coded genetic message. Right, right now you don't know. It is in the form of a code, AUG. What do you know? What is AUG? After translation, only we come to know that AUG stands for methionine, amino acid methionine. So this is translation is coded genetic message by messenger RNA is converted into polypeptide chain. See, on the basis of these message in the ribosome, what will happen? This will code for methionine. This will code for phenylalanine. This will code for valine. This will code for leucine, and this way on the basis of these amino acid finally will be will be joined by peptide bond, and what we will get, this is a protein. When many amino acid like methionine, phenylalanine, valine, leucine, they all attach by peptide bonds, we get a protein molecule. So finally protein will be peptide. That means the coded genetic message has been converted into a polypeptide chain. And what is this polypeptide chain? This is a protein molecule. And this with the complete process is over. This is all about, the central dogma is all about this only. Transcription, messenger RNA, DNA forms messenger RNA, and that messenger RNA which is carrying the coded message, that is decoded by the process of translation and is converted into a protein molecule or a polypeptide chain. This is the entire process of central dogma or translation. So not difficult, but when I do it stepwise, you will find it little, right now you have understood the process of translation, but when I'll start it stepwise, of course that is a proper way of learning stepwise. You will find it little, complicated, but that's not complicated if you know the mechanism, the introductory mechanism of translation. If you know that, then it's not difficult. So children, tomorrow we will start up with the stepwise process of translation, how it results in the synthesis of protein molecule. So right now, the time is over. I end the meeting for you all. Stay safe, stay healthy, children.